This week on Surfing the NASH Tsunami.
If an aspirin a day keeps the doctor away and it prevents you from having your stroke and your heart disease, then who cares about precision diagnostics to see if somebody needs an aspirin a day? I see the landscape of precision diagnostics with chronic liver disease and fatty liver disease, for which there are many integrated pathways and very heterogeneous outcomes, being no different.
Some patients are going to be a lot more expensive than others. Fast progressors will be more expensive. Patients with a high propensity to come quickly to HCC will be more expensive. So if precision medicine tells us which patients are at risk of imminent demise, and by the way, along with that imminent cost, then you start to figure out in a hurry, what do I want to do to treat the people who need the most help the fastest?
So in my mind, this is cirrhosis. I don't need much. But we sent her for a clinical trial. I got the biopsies showing F1. I mean, do I really believe this biopsy? I don't. Am I going to follow this patient as high risk? I will. So biopsy is not the ultimate decision maker for me because everything else is pointing in a different direction.
Last week, close to 175 stakeholders from across the global fatty liver community convened to hear 43 speakers address a range of NASH and NAFLD issues at the Fourth Global NASH Conference. Join hepatology researchers and key opinion leaders Professor Manal Abdelbalek, Doctors Naim Alkhouri and Ian Rowe, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and GenFit Global Diagnostics leader Suneil Hosmane as they review key issues and concepts that emerged from the Congress today on the Surfing the NASH Tsunami podcast.
For everyone with interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 2, Episode 24, our wrap-up on the Fourth Global NASH Congress here on Surfing the NASH Tsunami starts now. So the Congress itself was last Wednesday and Thursday. Many of you listeners know we had so much content in the episode on Thursday that we actually cut it into two and did not put out any conversations this weekend, but I want to thank everyone who's joined in. We've gotten over 600 downloads from people who came to hear our comments about the Congress, so that was a great thing, and we appreciate it very much. Also, we will be putting out a conversation tomorrow morning for those who are interested, a summary of many things that were said in all the different sessions on diagnostics, where I think a lot of the most interesting conversations went. Jorn is not with us today because he did the pre-meeting, but everybody else who was part of our coverage is here today. Naim, how are you?
Good. How are you, Roger?
Fine, thank you. I'm going around my screen in clockwise order. Louise, greetings.
Good evening.
Where are you tonight?
I'm at home. In London.
Okay. I saw the flowers in the background. I wondered if that was home or that was the yacht.
It's definitely not the yacht. They don't give me flowers yet.
No, I'm not going to ask which room you're in because Stephen isn't here to do the whole thing about your room. Welcome back. How are you?
I'm doing fine. Thank you, Roger.
Ian, welcome back.
Thanks very much.
And last but not least, Suneil. Hey, Suneil, congratulations on LabCorp launching NashNet today. That's a big accomplishment for you and for GenFit. Congratulations.
Oh, thank you. I really appreciate it. Actually, it happened on Friday. It was a little bit later in the day, and I'm glad I went out this morning. And yeah, big chapter. Of course, a lot of other stuff to do now, so it's not like the end of the road, but thank you. Appreciate it.
Sure. We weren't podcasting on Friday, so for us, it's all news today anyway. This is a really simple format. That meeting had so much content, and we had so many conversations, some of which were about the papers themselves and some of which digressed. There's a famous story about Kaufman and Hart, who wrote many of the Marx Brothers movies and plays. Being in the wings on Broadway, watching the Marx Brothers, who were notorious for ad-libbing, doing, I think it was The Night at the Opera, and at one point, Kaufman's in a conversation, he turns to him and says, wait a second, I think I heard a line from the script. We used those papers, I think, as a jumping off point, but the most interesting conversations I thought were not what was in the papers, but kind of where we went after we started talking about them. Here's what I'd like to suggest we do today. I'm going to ask everybody on the first go-around, just simple icebreaker, personal or professional best, either way, one thing that happened in the past week. Suneil, you can even go back to NashNet if you choose. And then after that, with Brave One first, I'm going to ask for one impression or one thought that you developed during the Congress and our discussions that you think is worth exploring a little more on the podcast, and we'll just take them one at a time and do them. With that, Brave One first, personal or professional best from the past week. Go ahead.
I'll go for it. We have this unprecedented situation in any disease state where you have so many diagnostic tests and so many many therapeutic agents, and we really don't have anything that's really mainstream. So if you look at diagnostics, I mean, in the United States, we have access to FibroScan, ARFI, shear wave elastography, MRE, corrected T1. You look at serologic stuff, FibroTest, FibroSpect, LiverFast. Now we have the NIS4, the NASH Next, which is exciting, but we really don't have any pathway that's agreed upon how to work up these patients and identify high-risk patients. And similarly with the therapeutic targets, I don't think we've had this in any disease state, in hepatology at least, where you had up to 70 potential, new drugs and nothing to compare to, no standard of care. Very exciting. I mean, I think for me, as someone involved in drug development, this is fun and exciting. This is the theme that I noticed, that so much is going on. We still need to implement this on a large scale in real life.
Thanks, Naim. Good place to start.
Well, I have personal. This past weekend, I took a road trip with my family from North Carolina to New Jersey to see extended family for the first time in 2 years. We'd all been vaccinated, and it was just incredibly liberating. And on the way up, of course, I was listening to the podcast. And the only thing that was running through my mind was, wow, I'm starting to see a light at the end of the tunnel. And if it's feeling this good for me, then certainly it's going to be so incrementally important for our patients with chronic liver disease, and really giving them accesses to care that I haven't been able to do as a provider for the past 12 to almost 18 months now. It was great to feel so liberated and to actually see family who I haven't seen in 18 to 24 months and feel a different sense of hope that I hadn't really sensed or felt prior to having my intimate and closest community totally be vaccinated, that we can gather together.
I'll go. I've got a personal one. We've now booked flights to get into Australia, so it looks like they're going to be letting us in towards the end of the year, which is excellent news, although it's on the proviso that they've done their vaccination program, which has hit a lot of stumbling blocks, shall we say, that they're not going to meet any of their targets. So whilst we'll be vaccinated, I'm hoping they will too. But from the conference, I was actually very struck by how much it's moving in different directions, from genetics and personalised NAFLD and NASH care all the way through to fitness and wellness, which I know Naim sings from very much the same hymn sheet as I do, in the fact that if we can prevent and find early location, you never need to be a liver patient. You may never need to be a diabetic either. So if we can do upstream as well as get better care downstream, then it's looking progressive, and particularly with all the NITs coming on board, we can use them throughout different specialities to really be more targeted and inclusive. So I was quite excited through the breadth of what was covered in 2 days.
Excellent. Excellent.
On Sunday, we managed the full set of results across 2 of our children's sports teams. A loss, which was taken very graciously, a tie, which was taken less graciously, and a win, which was celebrated a lot. So it was a whole cricket match and football match in one day for the older child was probably just about as much as he could manage. But it was great to be outside and enjoying a little bit of sunshine, although in March it's quite cold still for cricket here.
So how old is your older son, Ian?
He is 11.
I remember an old Nike campaign from a couple decades ago that said the best athlete in the world is a 10-year-old boy, and it was about how much running they do all day long that no one could ever catch up to once they were even professional athletes. Football and cricket in the same day, I think, would qualify.
I think he preferred to be a cricketer, but he would really, really love to be Andrew Robertson, who is the Liverpool left-back.
If he can't be back, he'd rather be him than Ben Stokes. Huh?
He's got great taste.
Okay. Suneil, who would you rather be, or what would you rather talk about?
I'll do a little bit of professional and personal. So the professional part is that launch. I think it's not so much a launch, but all the work that's gone into it over the years. You know, in full transparency, a long time ago when I worked at Intercept, we wanted to do something similar to this, just didn't have the program internally. And so to come like several years later to have it done, it's pretty nice closing of a chapter, I would say. Personally, I got 2 little ones as well, 2 little boys, and like they're getting more and more into sports, which is awesome. Nothing organized, but just going on the back and wanting to like dribble a basketball or like get on the bike and do stuff. I don't know. The weather's getting better. It feels great to be outside and being active as a family. So that's awesome.
So I'll do personal. I was with my grandson for a week, 3 weeks after he was born in July of 2019. And then when his mom brought him to Pennsylvania, I got to spend a day with him around Thanksgiving time. And I've not seen him since. And we made arrangements to go there for Father's Day over the weekend. And I'm so excited. I can't stand it. I think, Manal, I'm right there with you. This is on a personal and a professional level. It starts to feel that things are opening up. hopefully it will open up in a prudent way and we won't all find ourselves vastly disappointed as a lot of the European countries have. It feels good, I agree. So, as I said, theme of the day, pick an observation that you had coming out of the Congress that you think we can talk about in a thoughtful or controversial manner. Take your pick. And brave one, go first.
I'll go first. Overarching themes that I carried away from the conference is overall the field is moving closer to precision medicine. We are developing new technologies for biomarkers, which are discerning the underlying biology of the disease from RNA sequencing, the tissue level that is then subsequently detectable in urine or in blood, or even that correlates to other surrogates, breath testing even. And these markers are going to hopefully help us discern not only meaningful clinical outcomes as to who to really target for therapies and who we don't need to, but I find that it is opening up the door to our ability to tailor therapies accordingly for the pathogenic drivers of underlying disease. So when you take a look beyond the single abstracts, the unique data that's being presented, the overarching themes is the field is really moving very rapidly towards precision medicine. And I find that very exciting.
I'm glad actually you mentioned this, Manal, especially with the breath testing. I was excited to see this make a comeback. I worked on volatile organic compounds when I was at the Cleveland Clinic. We had a mass spectrometry machine. And this is a neat idea. I think, people don't understand it sometimes, but the idea is that, when you peel an orange and you have that smell, this is related to the volatile organic compounds that are produced. And we actually produce them in our breath on a daily basis. And we have hundreds of them that we can measure now with this mass spec technology. And we've published some results earlier on screening for fatty liver disease using volatile organic compounds. There's one called trimethylamine, which is a byproduct of the gut flora metabolism of choline, which is something that we eat in typically animal proteins and eggs, steak. And this is something that's actually upregulated or overproduced in patients with fatty liver disease. There's the whole story about endogenous ethanol production by the gut bacteria. Potentially, this is contributing also to NASH and progression of disease. And this is something you can measure in the breath. What's neat about the breath testing is that it's so easy. I mean, we talk about, how biopsy you cannot do multiple times, but this is even easier than blood work. I mean, you can do it once a week if you want to. Pretty simple. Obviously, it depends on what breath test you're doing, and some of them will require some preparation. We still have a lot to understand about how these volatile organic compounds are changed by diet and exercise and antibiotics and other things that we do day to day. But I think this is a neat idea, and I would love to see something that makes it to mainstream one day, because I think it's also attractive for children. If you're going to start early on identifying kids with fatty liver disease, that's the easiest way to do it, is a breath test.
Yeah, I thought the breath and the urine were really exciting. When this came up last week, one of the thoughts that accompanied it was about precision testing versus precision therapies, and the whole question of who can afford who, and frankly, which societies can afford to do what. As a way to understand disease completely, this stuff is fantastic, and maybe to understand it in individuals. I'm going to ask Louise and Ian to act like you work in a more resource-constrained healthcare system than Naim and Manal do, because you do. And I'm going to ask each of you as a group, the UK versus the US, and Suneil and I will moderate or throw bombs as we choose, to talk about how you think this will actually play out in your system and what's going to happen for it to have a real effect. Let me ask Louise and Ian to go first on that.
I think it's very difficult. We're very driven by evidence and cost effectivity. We're driven by NICE and review systems that decide a way, but a lot of it is still very siloed. There have been recent pathways for behaviour change that have looked at alcohol, that have looked at obesity, but not one of them linked into the NAFLD pathway. And when I raised the question of it was a missed opportunity because it was all about non-invasive technologies to look for liver disease, I was basically told that actually there's a NAFLD pathway, stick to the NAFLD pathway, but where we don't interlink it, we don't use the best opportunities to look at these patients. We spend more money in the long run. We don't resource the right areas because we're still fairly siloed. But I think just picking up what Manal and Naim was saying there, I think if we look at breath tests and urine, urine. And I know we have biomarkers currently, but we have a really big inequality in who accesses trials. We don't see people with disabilities accessing many trials. It's very difficult to biopsy somebody who's disabled and in a wheelchair. It's not the easiest thing to fibroscan, but those tests open up an equity to so many more and so much more of the populations that are affected the more we go non-invasive. There are massive opportunities, but are we going to grab them and make them the best? I'm not too sure in our system. Not the same as the VAs do that was discussed at the meeting, for example. They get access to a lot and they drive a lot of information. I love listening to their stuff because we don't have that here.
But Ian, do me a favor. Try to vision a path by which we get there from here.
So to try and work out how it would play in the UK, we'd need 3 critical pieces of information. I think there's work to do on each of those. So the first is about patient selection and about absolute risk of complications. And we talked a bit on Thursday night about using non-invasive testing to quantify risk across a scale rather than using biopsy outcomes. We might come back to that. But understanding the absolute risk of complications will allow you to understand what benefit you'll get with the therapy. But the other piece of information that you need to calculate that is the actual efficacy of treatment, and not efficacy against surrogates, but efficacy against clinical endpoints, because that ultimately will tell you how many life years you'll gain with therapy. There's also a sort of sub-question about quality of life, because that will play into the calculation of cost effectiveness. Once you've got all of that information, then the companies will know how they'll be able to price their drugs in the UK so that there'll be able to be some uptake. So if we had a very, very effective drug, a bit like a DAA, which I don't think is quite where we are yet, I guess we're more like, probably more in the sort of interferon, perhaps with ribavirin type arena. Those drugs, if they're used in populations who've got significant risk of liver-related death, meaning patients with cirrhosis or maybe F3, then that'll be something that might be cost effective, probably depending on the price then. To get to a point, if we were going to treat earlier in disease with lower absolute risks, either drugs have to be very, very, very effective, which I think will take probably multiple drugs because it's a multiple pathway disease. It's not like, a single virus etiology. So probably multiple drugs. In all reality, for the UK, where cost effectiveness is king, because that's how the market is governed, those drugs are going to have to be quite cheap. And, in that early stage disease, it's like primary prevention. It's lots of treatment that's effective in the population, but it's also cheap, so that you get all of the benefit across the population, but you don't break the bank in doing it. There's a real tension in trying to think about the development of personalised therapies in a disease that is highly prevalent, because it's a bit like going to your favourite boutique to buy your clothes, when actually what you need is cheap high street clothing for the masses. And until, at least for the UK, until that sort of tension is resolved to some extent, we're going to be stuck, I think, both conceptualizing treatment, but also companies might be stuck when they come here to think about how treatment might actually work and be a bit surprised by what the market really looks like.
Thanks. Those are both great answers. Naim, Manal, I didn't want to throw a wet blanket, but now I'd like you to come back and talk a little bit about how you see all this evolving in the States and where you see there being possible transferability.
I agree on several levels with what Ian has said. I mean, if you think about it, all of the excitement, need around precision diagnostics and precision therapeutics, but particularly precision diagnostics, is all going to be driven about their content of use. If we have therapies that are highly, highly effective, but yet carry with them concerns for safety signals or high health economic burden, there's going to be an absolute need for precision diagnostics. However, if you're to change that paradigm where you have a highly effective therapeutic target and it is incredibly cost effective with no side effects or risk, then who cares about precision diagnostics? Everybody should get access to a therapeutic without the precision diagnostics that is carrying us any farther than metabolic risk, obesity, bad on ultrasound, and liver enzymes. The need for precision diagnostics really depends on the scope and content of use for which the therapeutics is going to sit. If an aspirin a day keeps the doctor away and an aspirin's incredibly cheap and prevents you from having your stroke and your heart disease and even potentially even cancer, then who cares about precision diagnostics to see if somebody needs an aspirin, maybe aspirin a day? But that risk-benefit ratio completely flips if you're dealing with something like cancer oncology drugs, where you need exquisite genomic signatures for the disease to be able to tailor therapies effectively to get a efficacy benefit for which drugs either have high cost and a higher safety profile. And I see the landscape of precision diagnostics with chronic liver disease and fatty liver disease, for which there are many integrated pathways and very heterogeneous outcomes, being no different than what I've just defined for you, aspirin versus high-risk, high-cost therapies for which precision is incredibly necessary. So I really think to discuss precision diagnostics, the unfortunate yet the fortunate reality is both the therapeutics and the diagnostic pathways are happening in parallel, but we don't know what's going to be at the end of the race and whether there's going to be need for that level of precision diagnostics. It's ultimately going to be, in my opinion, dependent on the drug and the health economic risk profile of such a drug as to how the diagnostic space will unfold.
It's an interesting conversation. Some of it we won't know until we have data. For example, like oncology was not like that today. It was not like that, 20 years ago. It was, I kill everything in your body. I kill some things faster than others, and that's good enough. And that's first-line therapy. And then once we understood more, we unpacked the science, which means you had good trials and you start to uncover. Now you have microsatellite instabilities and MEK inhibitors and all this stuff. And that didn't come out overnight. That evolved. So, will NASH evolve to that? I don't know. So far, I don't think anything has given us a clear signal that that's there. But we also don't know as much about that disease, and you kind of won't know as much until you start routinely diagnosing people and you have a larger data set that's driving your insights. Today, I think it's a widespread population. I don't think there's anything that tells us precision diagnostics are needed, but I also don't think we know as much about the disease.
This is a hot topic, right? But I think we can discuss it on multiple levels. If we take it back to genetics and polymorphisms, something like PNPLA3, which we talked about, there's an interaction between the polymorphism and environmental factors and diet. There was a paper by Helen Hobbs and the group at UT Southwestern showing that if you maintain your BMI less than 25, even if you're homozygote for PNPLA3, you're not going to accumulate a lot of fat in the liver. As the BMI category goes up, you accumulate more and more fat with the same genetic polymorphism. Obviously, what Ian and Louise are saying in terms of targeting the population and healthy lifestyle, I think this is a first step in any intervention. But can we potentially, in a family where, 2 or 3 people already died from NASH cirrhosis or needed liver transplant, can we maybe run a genetic panel and see if we can identify PNPLA3 as a contributing factor? If we have a drug that can silence PNPLA3, target them with this, potentially, for some more complex cases. If we have medications that are powerful antifibrotics and we have patients with cirrhosis, even if they're, antisense oligonucleotides or short interfering RNA, can we utilize them in the tip of the iceberg, which is patients with very advanced fibrosis? I think there's an opportunity there. Again, on a population level, from a cost effectiveness, this is probably not the way to go, but we will have challenging cases where probably you need a little bit more precision. The other area is to try to find, what is the main pathway driving the disease. And so, in NASH, we have so many pathways similar to IBD, right? Crohn's disease. So you have these major pathways like the TNF pathway in Crohn's disease, and I've had several now TNF inhibitors that really made an impact. Can we identify in each patient what pathway is the major driver? And then if we have selective drugs targeted, can we actually look at some baseline predictors of response? So if I'm going to use an FGF21 versus an FGF19 versus resmetirom, is there anything I can measure at baseline that will tell me that probably you should go with this medicine first because you're more likely to show efficacy? I think we will get to that point. So many things to discuss. I think medicine in general is, proceeding toward precision medicine. The cost is coming down for genetic testing. And I think even drug discovery and being more targeted against a specific gene is becoming more and more feasible. Eventually we'll get there. But I think, we have to think about NAFLD. Is it going to be like type 2 diabetes management, or is it going to be like cancer management? Type 2 diabetes, we're not doing a lot with precision medicine now. Cancer, I think we are there. So we'll see how things evolve. But historically speaking, as you can can generate data cheaper and cheaper, and you have medications that can be more selective, I think we will get there at some point. So the question is when.
I'm thinking of a couple of commercial and economic issues. Number one, as night follows day, highly precise medicines lead to highly costly medicines. Nobody has yet made the decision not to do that. Okay, and part of the reason is when you say, is this like type 2 diabetes or is it like cancer? Type 2 diabetes is treated with less urgency and for more population. So, and I think NAFLD will look like that. If we look at what precision medicine can bring to the economics of disease, some patients are going to be a lot more expensive than others. Fast progressors will be more expensive. Patients with a high propensity to come quickly to HCC will be more expensive. If precision medicine tells us which patients are at risk of imminent demise, and by the way, along with that imminent cost, then you start to figure out in a hurry, what do I want to do to treat the people who need the most help the fastest? I'm not sure that's how the commercial equation is going to go, though. I don't have a window into it. In fact, of the 6 people on this line right now, I might have the least of a window into it, and I'm certainly not in the top 3. So I'm kind of interested in that.
For the next 10 years, Roger, it's going to look like the management of type 2 diabetes. I have no doubt in mind that this is the route we're going. After that, who knows what discoveries we make, what medications we have. We may change the way we approach these patients, but for now, it's going to look like the management of diabetes.
I've got a thought on what Manal was saying about aspirin. In the here and now, we know that we're targeting F3, F4, because that's where the highest mortality lies. But maybe Manal is right. Maybe we need to locate and prevent the fatty buildup right at the beginning. A simple, well, none of these drugs are simple, but a medication that keeps the liver fat low for those who can't use or don't respond to exercise, diet, and some of the metabolic responses. If you talk about the masses, we've got 16.4 million people here in the UK. There's 100 million in the US. If you talk cheap as chips, but mass volume, the prevention of long-term NAFLD and NASH particularly becomes a reality. If you develop, maybe simple steatosis is the target for the aspirin type medication to prevent the F3, F4 long term. We need to look at both scenarios.
This is where it comes back to absolute risk. So, I'm the wrong side of 40, although for those, nobody's actually met me except by video, so they can't tell that. The simple fact is that my risk of coronary heart disease event in the next decade is, I hope, quite low, less than a couple of % over the course of the next 10 years. But I wouldn't take an aspirin a day based on that risk, because there is a non-negligible risk of anemia, upper GI bleeding, and a few other nasty things that I prefer not to have. And for patients who've got steatosis only, whilst that's not benign in terms of its risks of future development of diabetes and perhaps the development of other metabolic comorbidities, the absolute risk is not sufficient to justify either the potential risks of a new and unproven therapy in long-term treatment, and indeed the costs. And this is where it comes back to, if you're going to have a drug that's going to work in F1, F2, think statin, not DAA. Think pennies, not hundreds of dollars. Only once you get into cirrhosis is the absolute risk of liver-related events sufficiently high that we're going to be in the realms of a even moderately costly therapy, at least in the cost-restrained NHS.
This all raises an interesting question, which is to run around from diagnostics to drugs for a minute. What was the most exciting thing you think discussed? And in that regard, we'll take into play now what the discussions were, what we each know in general. For the audience of that meeting, what was the most exciting or the most promising discussion about drugs or a drug or where things were going you found in that meeting? And if you were an attendee that didn't have the knowledge that, say, leading researchers have, what would you make of all that?
I guess the thing that piqued my interest most was the cyclophilin inhibitor, which is developed as an antifibrotic, and not just for NASH, but for all liver diseases, because that is the greatest unmet need that we have treating all patients. And if we could prevent disease progression from significant fibrosis to cirrhosis, and indeed reverse cirrhosis, then that would be a huge step forward for all patients with significant liver disease. And, there are many of those with unmet needs outside of the NASH space. I mean, I think if I was completely naive to the development of therapeutics in NASH, I would come out of the meeting thinking that there were lots of promising targets, there were lots of drugs in development, and that we will have a drug to treat patients with NASH and significant fibrosis quite soon, thinking maybe in the next 18 months or 2 years, hopefully. And I think that's quite different to the feeling that I might have got if I'd been to the meeting perhaps 12 or 18 months ago, where the feeling was less optimistic, I think.
I agree with Ian Ian's optimism, I think 2020, despite COVID and everything, it was a good year for NASH drug development. We saw the data with the semaglutide or NASH resolution close to 60%. We saw promising data with the PAM-PPAR inhibitor Inventiva, and Manal is one of the lead authors on that paper, where you had both NASH resolution and fibrosis regression, and that was significantly better than placebo in the high-dose lanifibranor. We saw the data, obviously, with resmetirom, and now they're enrolling, almost done for the phase 3, so hopefully we'll have these results. If everything goes according to plan, that would be probably the first drug in 2023. We have data on other agents, FGF19, FGF21. You know, when I used to talk to my diabetes colleagues about FGF21, just a few months back, they were not really too excited about it. They're like, it didn't really pan out in diabetes. To see all the potency and efficacy data now with companies like Akero, 89 Bio, it's looking very promising. I think not all FGF21s are created equal. You know, it has to do with the signaling pathways that they target and the receptors that they target. But, you know, this is one therapeutic target that I'm excited about, especially with the cirrhotic cohort with Akero. I mean, that was powerful, you know, regardless of how you think about it, small sample size, obviously limited study, but to see that regression in cirrhotics, that was very hopeful for me. So I think the pipeline is looking pretty good. Safety remains the biggest challenging question. We need more data on that. You can't really generate the data you need from a phase 2b, so phase 3 will be very revealing. We're looking at things like cardiac toxicity, bone toxicity, drug-induced liver injury is always on our mind. So a lot to talk about, but I am optimistic. I do believe 2023 will be the year we have the first FDA-approved NASH drug. I think patient selection also, we're getting closer and closer to saying goodbye to liver biopsy. My hope is that 2022 will be the year that we can say goodbye to biopsy, and then 2023 will be the year when we say welcome to the first NASH drug.
Wow, I agree with all that's been said. I render some pause only with the timeline. You guys are really ambitious, and the reason I render some pause and the head nod is while the drugs maybe show early evidence of promise and efficacy, I still think we have to overcome this hurdle of surrogate markers and endpoints from a regulatory standpoint. And I project your, 22, 23 time horizon for me is looking more like 24, 25. But maybe I'm struggling with, can we get there, because we still are grappling with the surrogates to the histology and to the ability to achieve registration endpoints. If we can't achieve registration endpoints, even though there's early proof of concept and even safety, we're not going to be able to cross this hurdle. And I've said this before, I think for biomarker development, we're validating these biomarkers against a surrogate. We're developing surrogates of a surrogate, and I think we have to potentially rethink this to develop a surrogate against the outcome, because we're using the histology as the gold standard by which to validate our non-invasive biomarkers. And I think that's one step removed from really the primary goal. So I see some regulatory hurdles here that I'd like to see fall by the wayside for us to get to that 22, 23 timeline that you're proposing. And I think it's going to be potentially a little bit beyond that.
My inclination would be to split the difference. I think the drugs might make it in 23. I can't see end of biopsy coming in 22. And Manal, you raise an interesting question, but I'm of two minds. If you can improve a surrogate biomarker by making it a more stable, reliable surrogate with less internal error to it, have you accomplished enough or not? And if the answer is yes, then I think we can get there from here without worrying about histology. The problem with ultimately having to lay up a surrogate marker against the clinical outcomes, given how slow the progression of the disease is that could be a decade or more. I think there's going to need to be some intellectual argument about, if you get a cleaner, more predictable surrogate, is that a better surrogate without even worrying about what it does to the endpoints, because we don't know what any of this does to the endpoints.
I think the data that came out of this Congress, and you can roll it into all the data that you've seen for NITs, but we always say that biopsy has some variability, it introduces a bias, and I think that's true. But I think we kind of know quantitatively what it is. If you look at even really sophisticated proteomics, multi-panel systems, right, where you're saying, I don't care about cost, I just want to get as close to truth as I can get. You see OROCs around 0.85, 0.86. I think you're asymptotically, that's the asymptote, I think, of what you can expect to achieve. I think 0.9 may be the highest you can ever do, and that 0.1 that you're missing is introduced through noise from the biopsy. So I think in some sense, we know how good we could get on that. That's my opinion. So we shouldn't try to do better is my point. I mean, if we're saying we need 0.95, that's hopeless. So be comfortable with what you can achieve. I think that it could be a multi-stage strategy. I'll give an example. I don't know if anyone's doing this, but you don't have to have the final answer. Like having correlation, association to outcomes, hard outcomes is where we want to go. It'll take time. We don't even have that for other trials, so that's a separate issue. But the FDA do accept progression to cirrhosis as a soft marker, if you want to call it that, right? We've seen with Stellar 3 as an example, they've used, you take your pick, you can take FibroScan, ELF, just for this conversation. And we know that they've built Kaplan-Meier curves, and they know that if you have a certain level of something, is it 11.25 on ELF, I don't know what the number is, that you have a higher risk of progression to cirrhosis as an example. And they've done something similar, I hope, with FibroScan. If you have that information and you've had that Kaplan-Meier curve built onto the Gilead study, all it takes is one or two other studies to replicate that in their study. Regenerate can achieve that. They have that data to support it. Madrigal can repeat that. They have the data to support it. Now you have at least enough data to support a conversation with the FDA. I think you can do it. I don't know if those groups are working in concert, but they could. So could you do it? I think you could. There's a lot of ifs there. Does the data replicate? Maybe it doesn't, right? In which case you got to go back to the drawing board. But if it does, you have a conversation that you can have. And I know the FDA is highly objective. And if you share that data, they're going to ask you certain questions. How did you enroll your population? Are there major differences? What did the data look like specifically? If you aggregate it together, what does it look like? But that's just my one view is taking sort of like this approach. It's not without its troubles, right? But I think it's doable.
I love what you're saying, Sunny. Roger, I know it sounds crazy to say we can say goodbye to biopsy in 2022, but what it takes is a leap of faith that we can do it without. If liver biopsy had higher complication rate, I promise you we would have moved away from it just to save lives, we're used to doing biopsies and liver disease, and this has been a limiting factor. So I'm just going to give you an example. Just today from my clinic, I saw a patient with the liver stiffness of 22 kilopascal on FibroScan, ultrasound showing splenomegaly and recanalization of the umbilical vein. Platelet count is 139,000. So in my mind, this is cirrhosis. I don't need much. But we sent her for a clinical trial. I got the biopsies showing F1. I mean, do I really believe this biopsy? I don't. Am I going to follow this patient as high risk? I will. So biopsy is not the ultimate decision maker for me because everything else is pointing in a different direction. Biopsy has issues. So you have to be open-minded that the gold standard is not great. It's invasive. We have a lot of data. I know FibroScan has issues, especially in the severely obese population. I know that we have issues with serologic markers. You need to understand the false positives, false negative, the disease prevalence, all of these things. But I think when I have multiple non-invasive biomarkers showing me the same thing, even if the biopsy doesn't support this, I'm not going to say, well, the biopsy is king and I'm just going to follow the biopsy. For this patient, she's high risk. I would treat her with medications. I think we just need to have confidence to say, hey, we've generated enough data. As Suneil said, if we have the data from Gilead, Intercept, Madrigal, that should be enough to show that these NITs have their own prognostic values. And I want to ask you, Suneil, a question. I mean, if we use your test, I think we have good data to predict fibrotic NASH at baseline. But what are the plans in terms of assessing response to treatment and prognosis? What data do you have and what data are you generating?
We have far more data on the prognostic question than we do for the monitoring, and that's only because all we have data is from Golden, and you need a true positive control, right? So I need to see movement, so I need to have more data to make that comment. But from a prognostic standpoint, I did exactly what I said here, which is I wanted to look at baseline stratification. So I know my patient, I stratify them based on score. Does that mean something? Is there a higher likelihood of progression over some fixed period of time, be it a year and a half? And the data that we had so far said that. Now, granted, we were looking at advanced fibrosis only because we wanted more N. We want to repeat it for cirrhosis, because that's closer to what we want the end goal to be. So the probability of progression, if you were in the highest risk strata, was around 50% in the course of 1 year versus 7% if you're in the indeterminate score zone versus 3%. So seemingly it's meaningful. Putting people in a different score strata means something, but we want to explore that and resolve it. Like I said, for anything else, like I want to see this replicated in data outside of our hands. I want to see it in independent studies, because then you know whether you have something real or is it an artifact of your data set. That's why we're so happy that this test launches, because we can begin to kick the tires on things and see where does it do well and where does it fail, and make the appropriate adjustments. So we've looked at other studies too. I think ELF has that capability as well. For that matter, FIP4 has that ability too, and NFS too. The only issues that we saw with NFS and FIP4 is you just miss a whole slew of patients. So when FIP4 puts you as high risk, you are high risk, guarantee that means something. It's very difficult to get into the high score strata, but many people who are high risk don't get put there. And that's the issue. How do you find all those people that you've missed? And I think that's where some of these other NITs can add value to what FIP4 and NFS are doing. They're not bad tests. They just need some support.
I mean, it's a fascinating conversation, really, because progression to cirrhosis is as much a surrogate as some of the other histological changes that we're measuring. And the prognostic value of an NIT at baseline in a trial, that predicts progression to cirrhosis, really probably just predicts cirrhosis at baseline. And it was the biopsy that was wrong in the first place. The proportion of true progressors, if you like, you could take Naim's patient that he described. If you biopsied that patient again, you'd be quite likely to find cirrhosis rather than F1, F2, or F3 the next time. And so if we're talking about validating a non-invasive biomarker against a surrogate of histology, then I think that might be quite a difficult conversation. I'm still not sure how much further it takes us from baseline prognostication. There are two different needs. One is in the clinician's hand. I want to know what the baseline risk for my patient is today starting treatment or entering surveillance for complications. And then there's the drug development need, which is I want a biomarker which will predict that this drug is doing something useful in terms of improving how a patient feels, functions, and survives. They are distinct issues, and I think we're there for the first one with almost without question, except for the issue that none of the non-invasive tests will accurately diagnose NASH. Good for fibrosis, but not so good for NASH. But in terms of prognostication against outcomes, I'm quite happy to use the test that I've got. Very rarely, if ever, need to do a liver biopsy to determine whether somebody's got cirrhosis or not, to try and understand what their outcome is going to be.
I think that's a really interesting answer, and it crystallizes a couple of things. Naim, the 2022 question is as much about regulatory agencies as anybody else, that the inertia of agencies is likely to rule out a 2022 walk away from biopsy, because the things that would lead them to walk away would be proof of outcome, and this is a slow-moving disease, or either markers or drugs available that will immediately overthrow the whole system. If the drugs aren't coming until 2023, and the new tests are not going to be able to provide that level of outcome guidance, because what are you going to measure them against if you can't get to outcomes fast enough? Then it's hard to envision overcoming the inertia necessary for regulatory agencies to get past biopsy that quickly. Manal, I don't know if that's what you were based on, but I know that was my thinking.
This whole discussion just fascinates me, and I gotta refer back, and I'm kind of smiling here because many of you who follow this field know Vlad Ratiu, and he was just so poignant in his depiction at this last NASH tag meeting of saying, we got to function like a cult. I mean, you're either going to believe that these non-invasive biomarkers are going to perform as well and move away and be part of the biomarker cult, and invest in them as a field in the same way that we've hinged and hung ourselves on biopsy being the be-all, end-all. But if we were to really hinge and hang ourselves on a different standard, we would allow ourselves to propel forward in a manner that it, in fact, winds up by sheer need and necessity and belief, replacing really as equal a surrogate in performance as many of these biomarkers we're talking about. He was so tongue-in-cheek that it got, a little bit of a chuckle. But in fact, he was very astute and I believe very poignant in his depiction of where we are at this crossroad between the biopsy and some of these surrogates, and really a relatively equitable misclassification rate when you think about it, given all some of the limitations, both sampling and interpretation that we otherwise have with biopsy. So I was reflecting on the discussions at the NASH tag meeting and thinking fondly of my colleague Vlad Ratio in his depiction.
Okay, this is the second episode in a row this has happened. Vlad's going to come on sometime in June or July and talk at length about making the transition from having been a believer that biopsy was what was going to happen to his halcyon cry to leave the cult. Couldn't agree with you more. And Louise is smiling because we were in an episode with him early in the podcast where that wasn't exactly where he was coming from. So we've watched this conversion happen in real time. Naim, we're at the end of an hour, so I think it's a good time to start to wrap up. Let me do one final question. To people who aren't researchers, but people who treat patients, kind of lay treaters, the one thing you would advise that they take away from that Congress and our discussions that they might think about doing differently or looking at differently, what would that one thing be?
I mean, I think it'll be to look at all these diagnostic biomarkers and pathways and develop their own pathway depending on what they have access to. So if they practice in an area with very limited resources, even implementing something like the FIP4 as a first step, I think this would be really important for primary care physicians, family physicians, at least to think about staging the disease and trying to identify high-risk patients. Just looking into the resources available to them because, many radiology departments have technologies to measure liver stiffness. They don't advertise it. So sometimes the providers are not even aware that they implemented RFI on their ultrasound machine. It doesn't have to be always the latest and greatest technology. I think this would be the message for people who are not heavily involved in research. And just to go back to the biopsy, just the last thing, myself and Manal and Vlad and our friend Mazen Nuruddin, we're doing a webinar through ASLD on challenges in trials for NASH, including the issue with histologic challenges and how we can utilize NITs to design trials without biopsy.
Do you have a date for our listeners who are ASLD members? Do you have a date for that?
Yes, it's May 19th, and it's a webinar, and I think it's free registration, so anyone can register and listen in, and we'll take questions. So, I highly encourage you to attend and be provocative and challenge us on the points that we will be making. But the premise of my talk is that we need to think about alternatives. I don't want to give away the punchline, I guess, so stay tuned.
We will. And thanks for being with us today, Naim. That's great. Okay, who wants to go next?
I think COVID has changed the landscape, obviously, for everybody. And I think if I wanted people who were not hepatologists to take away anything from what we're talking about in this meeting in non-invasive technologies, biomarkers, is actually, and particularly in the UK, to demand access to them. I think we have very limited access outside hepatology specialist units to things like FibroScan, to a lot of the blood tests. A lot of primary care physicians don't do full blood screening for liver disease. They don't do ALT, AST, so we're missing part of the ability to use FAST if we had it. And I think the time for endocrinology and cardiology to start ruling out fatty liver disease as a risk in their population is coming to the fore post-COVID with all of our joint patient comorbidities. And I think people need to now demand access to non-invasive technologies which are cost-effective and become more cost-effective the more access they get to them. So that's what I'd want people to take away. They're available. Let's start using them.
Well, if I go clockwise on my screen, and now I land on you.
Yeah, I think we just need to keep on keeping on. The field is evolving. We need everybody who's involved to be committed in doing their due diligence. It astounds me how much progress we've made, even despite COVID. So the field hasn't been stunted and it hasn't been stalled. So I'm excited as to, while I think it's optimistic, the time trajectory that has been proposed in the upcoming 2 years to see some really innovative switches to diagnostics and therapeutics available.
Great. Thank you so much. Ian?
I think to return to what Louise and Naim have said, it's probably doesn't matter what staging non-invasive test you do, just do one. We're probably quite close to the ceiling of their prediction against liver biopsy in terms of global performance. So it doesn't matter what you do, identify those patients at high risk, but don't having done that, don't neglect their comorbidities and make sure that they're managed.
Okay, good. Suneil?
Oh, I guess I misinterpreted. I felt like I'm not in the position to make any comment or recommendation.
I'm not either, but it's not going to stop me. So I'll make one, and then if you have one after that, we'll come back to it. I would like to suggest that we try to divorce diagnostics from drugs, that I believe one of the reasons that people aren't motivated to diagnose more aggressively is because they ask, in the absence of drugs, what is there to do? And the two papers from Suzanne Norris and Yashira Zelversagi, I think, made the point, as Louise has emphatically to passionately, as long as she's been on this podcast, that there is much to do. Many patients can self-treat if, in fact, you give them the right tools to self-monitor and self-motivate, that people will do better if they know they need to do better. And we shouldn't let the idea that lots of people only want a pill, and we can't give them one right now, to get in the way of doing the diagnostics that are available. I think divorcing those two things will be the greatest single motivator to get practitioners to use non-invasive tests, because now they know why they're doing it, as compared to simply for hypothetical. So to the degree to which we can separate those two things, I think will be what will help to achieve the goal we're all talking about here, which is make non-invasive testing the standard by which patients are evaluated. And that would be my advice. Suneil, you got one?
I actually agree with everything, but I think it's just pick something and do it. Just do something, like rather than, try to boil the ocean for the best recipe and all of that. I think that all comes with experience, right? So unless you start using things, getting a feeling for them, you're not going to get better at anything. And I also agree, just from a patient perspective, like sometimes you may have a sense that something is wrong, or you may have a sense that you're not feeling well. It's very, very different. I mean, for those of you who have an Apple Watch and you use it, right? You know when you've been lazy and when you've been sitting, but when 7 p.m. rolls around and you see none of your rings closed and you're like, oh my God, what have I not done today? It sometimes can light something, and you may go out for that little run or that little walk and try to do something. So in that light, information's very powerful, and if you just do something and can communicate that to your patient, it'll start a series of events, and you won't get everyone, but if you can touch just even a handful of patients, that's improvements. So
amen to that. And with that, let everybody go. I'm going to do a little bit of business on the end of this podcast, but y'all don't need to stick around for that. So I want to thank Jorn, who isn't here today, and Naim, and all of you for being part of this coverage. I think we've brought a real benefit, but based on the feedback we've gotten and the messages we've gotten, I think we brought a real benefit to people, and help them process and think about this meeting and what there was to learn. So that's what we're here to do, right? And that's worked out pretty well. With that, thanks all of you. I'm going to cut off this recording now. Please don't jump off until you're allowed. Thanks.
We don't have very much business to discuss this week, but here goes.
On to our business for the week. Actually, we don't have a lot of business this week. I want to thank Global Engage for everything that they've done to help us pull together coverage of the Global NASH Congress, and the several hundred of you who have checked in or downloaded material from that meeting, and the panelists who did such a great job of getting all that together. Beyond that, next week we will be back with original content episodes. The month of May has some promising things in it. We'll begin previewing EASL towards the end of the month. We will be talking about NAFLD epidemiology at some point this month, and we will be back with Mazen Noordine on the 17th, talking about the question of how do you de-escalate coverage and what would a treatment pathway in and out of NASH look like. That's it for today. We'll be back next week on Wednesday, May 12th. And beyond that, let me just say thank you to everybody, to Mike Wilson, who makes all this sound so good, to Eric and Polly for everything they do, to you, the listeners, who stay with us and keep making this worth doing and fun and vital. So until next week, everybody just take care, stay safe, surf on, and we'll see you on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to questions@surfingnash.com, and we will answer on episode 25 or on the website. Join us next time on Wednesday, May 12th, when Stephen Harrison returns to the podcast for another fascinating episode, right here on Surfing the NASH Tsunami.